Hello, and welcome to this video on neurodevelopment, which is the development of the brain and the spinal cord. And if you are watching and understanding this video, it's very likely that at some point your brain and spinal cord did in fact develop. This is a rather commonplace occurrence for everyone. And yet, if you think about it, neurodevelopment begins with a single fertilized egg and ends with a functional adult brain. And so even though this is a rather commonplace occurrence, it happens for everyone, it really is a remarkable procedure. How does this happen? What are the processes that allow this to occur? So there are three main goals of neurodevelopment. The first is that cells in the nervous system must differentiate. We know that there are different types of cells in the brain and also throughout the body. For example, in the cerebellum, there are Purkinje cells. There are also retinal ganglion cells, each of which has a different structure and function. And so during neurodevelopment, the cells must differentiate into these different types of cells that will support their function as well. Second goal is that cells must find their way to appropriate sites. So once they've differentiated or started to differentiate, they must find their final location. And then the third goal is that cells must establish connections with other cells in a way that will be meaningful and allow them to function properly. So we call this synaptogenesis, or the creation of synapses. And all three of these goals are important for the brain, for the nervous system in general, to function properly. And so with this lecture series, we will devote one part to discuss each of these three goals: the differentiation of cells, the migration of cells to their appropriate sites, and then also the synaptogenesis, the creation of synapses or connections with other neurons so that they can function properly. And these goals correspond with 5 phases of neurodevelopment, and we'll speak about each of these in turn. First of all, the induction of the neural plate and neural proliferation or multiplication, division of neurons. Migration, as we talked about, those neurons need to find their appropriate sites. The axon growth and synapse formation, that refers to forming those connections between the developing neurons. And while this may be surprising, neurodevelopment also includes a phase of death of neurons and rearrangement of the synapses. So just because a neuron has been differentiated, it's migrated to its specific site, it's grown axons and formed synapses, there can still be some modulation related to neural development. So we'll start with this first phase where the embryo implants and starts to differentiate into the epidermal, or kind of the skin layer, and also the neural precursor cells. Before these cells become neurons or glial cells, they are neural precursor cells. And these neural precursor cells form what is known as the neural plate, which I'll show you in a moment. And this neural plate then folds to become the neural tube and neural crest cells. And so this is an important stage of neural development. So the fertilized egg first undergoes cell division until it becomes a blastocyst, which is a cluster of these dividing cells made from the fertilized egg. This blastocyst is implanted into the uterine wall, and it differentiates into a trilaminar embryo, meaning that the embryo has 3 different layers, or we call them germ layers. There is the ectoderm, which then goes on to develop into the nervous system and also the skin. the mesoderm, which later forms the muscle cells and connective tissues, and also the endoderm, which develops into the digestive and respiratory systems. So you see here in this figure, in this micrograph, the ectoderm shown here on top. And again, the nervous system derives from the ectoderm. Below that is the mesoderm, which develops into muscle cells and connective tissues. And then on the bottom shown here is the endoderm. And initially, there is this neural plate, which is shown here at the top. And you can see these three germ layers. The ectoderm, again, shown on top, the mesoderm, shown here in blue, and the endoderm, shown here in brown. The neural plate then begins to fold up to form the neural tube, which then becomes the central nervous system. The neural crest cells at the top here later develop into the peripheral nervous system. So the neural tube later becomes the central nervous system. The neural crest cells develop into the peripheral nervous system. And so you can see in this figure down here, we are looking down on this neural plate. It shows the rostral end and the caudal end. The rostral end, this is what develops into the brain. The caudal end develops into the spinal cord. And so looking here at a cross section, we see that it kind of looks flat-ish. It kind of looks like a plate, so the neural plate. Over time, this the neural groove develops. So it starts to kind of fold a little bit, and it develops this neural groove. Again, looking here down from the top, and then cross-sectionally from the side, you can see that neural groove developing there. And then with additional time, you can see over here that this neural tube has now formed. And again, from the cross section, this shows the roof plate. This shows the neural tube formed here. And then there's the floor plate, which we'll talk about a little bit later. Over here in this column, we see the same thing, but it helps to see the actual images of what this looks like and how they correspond with these cartoon drawings. So again, starts with the neural plate. That neural groove starts to form, and you can see how that corresponds with the micrograph on the right. And again, you can see a little bit better here, those three layers, the ectoderm, the mesoderm, and the endoderm, as this neural groove starts to form. With additional time, the neural tube starts to close, and we can see how that corresponds with the image here on the right. There's that neural tube. And then down at the bottom, we again see this neural tube. And on either side, we now have the formation of somites. And these are precursor populations of cells that will become the skin, the cartilage, tendons, vertebrae, and so forth. And again, you can see how that corresponds with the micrograph there on the right. This is another view showing the same thing here on the in A on the left. This shows where the neural groove is still open, and then in B, this shows that that neural groove has closed. And you see these little round or spherical structures along the sides. These are those somites, which later become the skin and cartilage and so forth. And so by about the fourth week, this neural groove has closed. Now, when this neural tube does not close appropriately, it corresponds with neural tube defects. If the anterior of the neural tube does not fully close, that results in what is known as anencephaly, or the failure of the brain to form. and that is typically not compatible with life. If the posterior of that neural tube does not fully close, that is associated with spina bifida. And there is a video here. You see the link down below that you can watch to learn more about these neural tube defects. And obviously, during pregnancy, we would typically want to prevent neural tube defects from occurring. And as a result, folate supplementation is often recommended during pregnancy in order to prevent neural tube defects. So once this neural tube has formed, the cells of the tube then begin to proliferate. They tend to multiply. And most of this cell division occurs in the ventricular zone, so near the ventricles. And the pattern of this proliferation is controlled by chemical signals. And transcription factors are really important for this neural proliferation. These transcription factors are proteins that bind to DNA and activate or repress certain gene expressions. And during neural development, these transcription factors dictate the fate of different neural precursor cells. So even though the cells all have the same DNA, it's these transcription factors that will help determine what kind of neurons these precursor cells become. And there are two types of neural patterning. There is the dorsal ventral patterning and also the rostral caudal patterning. So the location of where these neurons are within the developing brain are very important because it helps to determine what types of cells they become. Now, rostral versus caudal. As a reminder, in the brain, the front of the brain is rostral, and the back of the brain and down the spinal cord refers to the caudal direction. In the brain, dorsal refers to superior. So the top of the brain is the dorsal brain, whereas the ventral brain is inferior. Now, in the spinal cord, the dorsal spinal cord is towards the back, and the ventral spinal cord is towards the front. There is this curve that occurs right about the midbrain, where that direction changes in the human brain and spinal cord. So again, rostral towards the front, caudal towards the back, dorsal towards the top, ventral is more inferior. And so when we're talking about dorsal ventral patterning, this would be the top of the neural tube, and this is the bottom of it. At the roof plate or the dorsal side, this is what releases BMP. And BMP stands for bone morphogenetic proteins. It's a family of proteins that are similar. And then at the floor plate of that neural tube, above the notochord, this is the ventral side of that neural tube, and it releases sonic hedgehog, which you might sometimes see written as SHH, an acronym for sonic hedgehog. Now, these are signaling proteins that help to determine what the different cells become. So the cells that are close to the roof plate that sense a lot of BMP, these become sensory interneurons. Cells that are close to the floor plate and sense a lot of sonic hedgehog become motor neurons. Cells that are further away from the floor plate that don't sense as much sonic hedgehog, those become the motor interneurons. So we already have this patterning where cells that are dorsally located, those become the sensory neurons, whereas those that are closer to the floor or ventral those become the motor neurons. And there's a similar type of patterning that occurs rostrally versus caudally in the developing brain. This neural tube differentiates into different structures, the forebrain, the part that we typically think of as the brain, the midbrain, the hindbrain, and then the spinal cord. The forebrain differentiates into the telencephalon, the cerebral hemispheres, and the diencephalon, which includes the thalamus and the hypothalamus. So you see that shown here on the right. This is the 5-vesicle stage shown here at 5 weeks. These will become later on will become the cerebral hemispheres. And here we have the precursors to the thalamus and the hypothalamus and so forth. The midbrain here includes the cerebral peduncles, the midbrain, tectum and tegmentum that will develop later on. The hindbrain later becomes the pons and the cerebellum, as well as the medulla oblongata. And then down below, this is the precursor to the spinal cord. This is, again, showing some of those stages of neural development. This represents embryonic day 19. And at this point, we again see that neural fold forming. And by E20, kind of starting to close a little bit more. And then down here below, we see kind of a side view of this developing brain. So we have what will later become the forebrain here, the midbrain, and the hindbrain. And by embryonic day 49, you can see those differentiated a little bit better. So we have the telencephalon developing here, the diencephalon, the mesencephalon or the midbrain, the metencephalon, so the pons and the cerebellum, and the myelencephalon, that medulla oblongata, and then down below, more caudally, the spinal cord. And so at this point, we've talked about the formation of the neural tube and the differentiation of the different types of cells that will become neurons. And in the next segment, we'll talk more about the migration, or how those cells get to their appropriate place so that they can function properly in the developing brain.